Welcome everyone to Alumni Week and tonight's Kraw Lecture. I'm George Kraw here with Raphael Kraw. The Kraw Lecture series features acclaimed UC Santa Cruz scientists and technologists who are grappling with some of the biggest problems of our times. In recent years, the use of DNA has been the focus of real crime podcasts and crime documentaries. Shedding a light on these powerful new technologies that are helping to solve these crimes are tonight's topic. Tonight, we will bring you a discussion about the forensic genetic genealogy approach for identifying suspects in violent crimes and the new technologies behind these advances. Moderating tonight's discussion is Alexander Wolf, who serves as Dean of the Baskin School of Engineering and is a distinguished professor in the Department of Computer Science and Engineering at UC Santa Cruz. Professor Wolf received MS and PhD degrees from the Department of Computer Science at the University of Massachusetts Amherst before joining AT&T Bell Laboratories. He was a professor at the University of Colorado Boulder, a professor at the University of Lugano in Switzerland, and also at Imperial College in London, United Kingdom. Professor Wolf's research interests span the areas of distributed systems, networking, and software engineering. He is a fellow of the Association for Computer Machinery, IEEE, and the British Computer Society. He's also a member of the Computer Research Association Board of Directors and the American Association for Engineering Education Public Policy Committee. Before we begin, I would like to share a few details about tonight's event. We are using a webinar tool, so there is no chat function. We will have an opportunity to answer your questions later in the program and at the end of the program. And we invite you to submit your questions into the Q&A box at any time. Tonight's event will be recorded and available later at the university's YouTube channel. Now I'll turn this over to Dean Wolf.
Thank you, George, and thank you, Ray, for hosting this wonderful series of events. And we've had the opportunity to participate in them a number of times now, and I'm really looking forward to tonight's event. I want to thank the event organizers who do such a wonderful job putting these together. And of course, I want to thank tonight's guest, Professor of Biomolecular Engineering, Ed Green. And I will be telling you more about Ed in a moment. Ed is actually part of a rather sizable group of faculty at Baskin Engineering that span our academic departments who are involved in innovative and often life-saving research in the area of health engineering. This is groundbreaking work that we have taken up in the School of Engineering, and you will probably know the work that we've done in computational genomics and nanopore sequencing, which really put Baskin Engineering on the map in the health engineering field. More recently, we've become very engaged with COVID-19, of course, taking up that challenge. Some of that work is actually presented in Kraw Lectures. Holger Schmidt and David Hausler both gave lectures in the Kraw series this fall. And actually a year ago, Rebecca Dubois gave a talk on virology and the relationship to the pandemic. So this was very early on in the fight against COVID-19. We're doing other research, of course, just a sample of that in health engineering. Angela Brooks is working on new approaches to treat cancer through RNA analysis. This was also the subject of a Kraw lecture back in 2019. Our faculty are working on innovative subjects such as wound healing, severe wound healing. And this is a collaboration between our biomolecular engineering, our applied mathematics, and our electrical and computer engineering faculty. They're using bioelectronic devices, machine learning, and regenerative medicine to heal really severe wounds in, as you can imagine, in severe cases. We have a recent award to Sophie Salama who is studying molecular and cellular defects that underlie complex neurodevelopmental diseases like autism and schizophrenia. Not only are faculty involved in this kind of innovative work, but our students are also deeply involved. We have our participants in the annual iGEM computation, which bring together multidisciplinary teams to work on the design, the building, the testing, and the evaluation of systems in synthetic biology, solving real-world problems in a project setting, and then competing actually in an international event. This year, the UC Santa Cruz iGEM team came up with a gold medal in that competition. So we're very proud of their work. Now, let me turn to Ed. Ed is a paleogeneticist. So paleo as in ancient or dead, you might think of it. And that'll become more meaningful as you listen to the talk tonight. Ed holds a PhD in molecular and cell biology from Berkeley. He has a BS in genetics from the University of Georgia. And he served as a Peace Corps volunteer in Eritrea after graduating from college. From 2015 to 2010, he held a postdoc as an NSF fellow at the Max Planck Institute for Evolutionary Anthropology in Leipzig, Germany, after which he joined our Department of Biomolecular Engineering. He's published papers in the Proceedings of the National Academy of Sciences, Nature, Science, and numerous other prestigious journals. He was awarded the Newcomb Cleveland Prize by the AAAS in 2011, the association's oldest award. Ed is also very engaged with the community. He has been active in speaking and visiting with various members of our community. He's given lectures at the Exploratorium, San Francisco, Café Scientifique in Silicon Valley, local retirement communities, and also the Access program at Cabrillo College, which is our local community college here. Aside from all this, Ed is an avid entrepreneur. He's founded 3 companies: Dovetail Genomics, which started in 2013, Claret Bioscience, which is innovating in next generation sequencing technologies and DNA assay design, and Astrea Forensics, which uses paleogenomics to help law enforcement solve cold cases. As it turns out, the CEO of Astrea Forensics and Claret Bioscience, Kelly Harkins Kincaid, was the guest speaker last evening in season 3 of the Diverse Voices speaker series in Baskin Engineering. Diverse Voices brings entrepreneurs and tech professionals from traditionally underrepresented groups to talk to our students about their path to success. And Kelly, as the CEO of these 2 companies, gave a wonderful, wonderful presentation about her pathway and her work, as it turns out, with Ed. Kelly was actually a postdoc with Ed at one time in his paleogenomics lab. So the connections there are very, very strong and meaningful. So tonight, Ed is going to be talking about work that's based on what he is really very, very famous for in the scientific community. It's based on the work he did on the Neanderthal genome. His team sequenced the mitochondrial DNA that were found in a 38,000-year-old bone fragment found in Croatia. The work led to breakthroughs in our understanding of the evolution of modern humans. The technology that Ed is going to talk about today, described in a New York Times article as a game changer for crime and surveillance, makes it possible to recover and sequence DNA as a forensic tool in criminal investigations. Ed is somebody who really enjoys interaction, so I would invite you to put any questions you have into the Q&A into the Q&A slot. I might interrupt Ed and ask a question at some point. Ed is going to be taking a short break in the middle where he will take a series of questions. Ed said that he really enjoys provocative questions, so please be a provocateur. So without further ado, Ed Green.
Well, thank you very much, Dean Wolf, and thank you to the organizers and George and Rafa Kraw for organizing or making all of this possible. I'm going to share my screen now, and this is going to work flawlessly, I hope. Let's see. Okay, so as Dean Wolf mentioned, the talk that I'm going to give tonight is about DNA-based forensics. My lab works on DNA technologies in many arenas. We work on a lot of ancient DNA or paleogenomics things where the technical challenge is simple in concept and hard in practice. The concept is you've got a really, really old sample with tiny amounts of fragmented DNA from a bone that's 10,000 years old or 20,000 years old or some really crappy sample, and you want to get the DNA out and you want to get it converted into a form that can be sequenced. And for a decade now, we've been thinking about this problem and developing technologies to address it. It turns out that the field of DNA-based forensics has a lot in common with ancient DNA. Often there are small amounts of crappy samples and you want to get the DNA out and do some interpretation of it. So it was maybe a natural thing that eventually I would go in this direction. I don't know, I'm gonna move that a little bit. But what really got me interested in this and got my lab into it was a case that I will tell you about here because it's kind of interesting. This is how my lab got into DNA-based forensics. And it's the case of this casket that was dug up underneath the house of a family in San Francisco in the Rossi Street Park neighborhood of San Francisco. They were out of town. A couple was out of town and some work was being done in the basements, and you can see the post hole being dug there, and the workers, when digging, hit something, pulled it out, and it turns out it was a casket, and not just any casket. It was a casket that kind of looked like it was designed to be as creepy as possible when it would be disinterred many years later. It has glass plates on it, so you can see directly into the casket, and the workers who dug this out could see, you know, the little girl in there as soon as they got this casket out of the ground. It was a steel casket that could be sealed in an airtight way. Really, really strange. They get on the phone and call the couple who was out of town while this work is being done, and you know, ask them, "Is there anything you want to tell us? We found something in your basement, underneath your basement." And of course, they had no idea what this was about, and started an odyssey to try to figure out why would there be a casket with a little girl in the casket buried underneath our house in San Francisco. And more or less got nowhere with the city. The police didn't want anything to do with it because there was no indication that there was a crime. The coroner, for various reasons, was no help. And in just looking in the internet, Googled, what do I do with the body of a dead kid? They happened upon this lady, Alyssa Davey, who is out of San Diego and is a real live saint walking on earth now. She runs this organization called the Garden of Innocence, and their mission is to provide dignified burials in the case where a child is found, a dead child is found, and no one will claim the child. Evidently, this is a thing that happens, and she says, my group, we'll take the child and make arrangements for dignified burial. So the couple called Alyssa Davy, described their situation. Alyssa Davy said, this is not normally the kind of situation that we deal with. Usually there's not a casket, but sure, I will help you. And Alyssa Davy, in a very forward-thinking way, assembled a group of folks, anthropologists, historians, genealogists, and us to work on the DNA to try to figure out who this girl was. And this was Teen Miranda Eve, because the family that lived in the house, there were two little girls who lived there. And when they were told that another little girl was found buried underneath the house, they claimed they knew she was there all along and in fact seen her, and her name was Miranda Eve. Kind of creepy, but that was lacking a name. We just went with Miranda Eve. So we took a hair sample. We didn't want to do any kind of destructive sampling of the body, but took hair. We've known for some time now that it's possible to get really fragmented DNA out of hair, and took a lock of hair, cut the hair to our lab for the DNA work. And as I said, it's been known for some time that there is DNA in hair. It's variable, the amount that's there. And as I'll get into in the second part of this talk, we've learned enormous amounts about the characteristics of DNA that is in hair. But for now, suffice it to say that we know there's DNA in hair. We can look at it in the microscope. You can stain DNA and permeabilize the hair, and then look in a microscope and see that the DNA is present. We'll get into this a little bit later, but the point here is that the length of the DNA fragments, whether it's from the nuclear genome or the mitochondrial genome, they're very, very short. This is from fresh cut hair. The DNA that we got from Miranda Eve, when we sequenced it, we got it out, converted it into a library using the technology available to us at the time. We saw that there was this very kind of characteristic pattern that we see in ancient DNA, where at the beginning of each DNA fragment, there was a lot of chemical damage that causes the cytosine, one of the 4 bases in DNA, to be misread as a thymidine. And the reverse of this at the ends of molecules, the guanosine to be read as adenosine, so G to A at the end, C to T at the beginning. This signal is something that we know we're very familiar with. This means ancient DNA. This is a chemical damage that accumulates over many, many, many decades. And seeing this very clearly let us know that this is a very old sample. That girl has been in the ground for a while. And the length distribution of the fragments, how long the pieces of DNA were, are even shorter than what we see in fresh cut hair. So that's consistent also with the sample being very old. We were able to get a full mitochondria from the sequence data that we got. Her haplotype was an I haplotype, which is most common in the British Isles. And overall, we amassed about 13% of the genome. So most of the genome we didn't see from the data we had, but about 13%, some coverage here and there we can see of her genome. And in the meantime, while we're doing all this, we watch the historians and the genealogists, and those guys get to work. And here's what they found. This is a picture of the Rossi Street neighborhood in 1899 in San Francisco. And the first giant clue of what's going on here is this. There was a cemetery there. That neighborhood used to be a cemetery. And so with that giant clue, we start trying to figure out, or they start trying to figure out, okay, well, what happened to that cemetery? That is known. This cemetery and all cemeteries in San Francisco were fully exhumed or nearly fully exhumed. The impetus for this really grew out of the earthquake in 1906 when the markers, the grave markers, were knocked over and moved around, and it was difficult to tell who was buried where anymore. And after that, and because of the increasing population and insatiable demand for more and more land for for people, it was decided that all of the cemeteries would have all of the bodies exhumed and moved down to Daly City. That's why there are no longer any cemeteries in San Francisco. This is a picture of that activity. You can kind of see in the distance here in this picture, the domed roofed building. That's the columbarium. It still stands in San Francisco today. And in the columbarium are the cremains of people. You can rent a little cubicle there and have a little diorama of your life and leave your cremains there if you want your remains to be in San Francisco, but you can't be buried there anymore. So looking more at the records of who might have been buried in that cemetery and who this girl might have been, they found this map of the Oddfellow Cemetery and the paths that used to be there. And looking at other records, they could try to figure out which families were buried in which sections of this cemetery to come up with a hypothesis about who this girl might have been. From this map and this really key picture, we were able to get some hypotheses delivered to us. And this picture wound up being so valuable because you can see kind of in the distance here, I hope you guys can see my mouse. If not, it's kind of a little bit to the left-hand side of the screen and a little bit up from the middle. The paths of the Rossi Street, or excuse me, the Oddfellow Cemetery in what will become the Rossi Street neighborhood. And you can see the columbarium there as well. You can also see in the distance the two towers of the Golden Gate Bridge, but there's no span yet between them, which lets us know in time when this picture was taken. This picture was taken, the story is, in preparation for Amelia Earhart flying across San Francisco. They wanted to get a picture of that from the air, and this was a practice picture of that. Turns out to be extremely valuable to try to figure out who this girl is, because we can, of course, make a similar view now from Google Earth of what San Francisco looks like today, or at least looked like at the time we were doing this project several years ago, and try to figure out exactly where that house was, exactly what path that was on, and which family might have been buried there. That narrowed it down to 3 individuals who were about the right age. Turns out that in the distant past, children died much more often than they do today, and the cemeteries of the past had more kids in them than cemeteries do today. But narrowing it down to 3 possibilities was, from all the possibilities that existed before that, was an enormous help. Now we're down to cases, and we can go into individually investigate each one. One of these, the one I'm going to talk about because it turned out to be the right one, was this. Edith Howard Cook was a little girl who lived from 1873 to 1876. She has a living relative today, a fellow by the name of Peter Cook, who I had the pleasure of meeting when we took the DNA sample, got him to submit a spit sample to get DNA out of. And now this kind of wide open question of who is this girl can be focused into a very specific question. Is that girl the great aunt of Peter Cook? And if so, that girl is Edith Howard Cook. So is Miranda Eve the great aunt of Peter Cook? That set us on this path of technology development now to answer this very specific question. When one has 13% of a genome, and one can then get as much DNA and as much DNA sequence as one wants to a comparison individual, how do you do that? How does one answer this question? Let's see. I saw there was something on the chat. If someone needs to say something to me, I can't access that. Just shout it out. Otherwise, I just keep going. So we developed some technology now to identify what are called regions that are identical by descent, regions of the genome that are exactly the same over long distances as you get between people who are very close relatives. And to address exactly this question, And on the upper left-hand corner here is shown the results of this for a positive control. These are two samples, the NA12890 and the other one are a grandmother and granddaughter. You expect about 25% of the genome to be identical by descent there. And the blue segments are the ones that are confidently called identical by descent. In the bottom left is a negative control. This is Miranda Eve, our sample, where we have very little data, compared against a random person from a publicly available database who happens to be from Great Britain. As you can see here, there's one little segment where it looks like there might be some cryptic relatedness, but mostly unrelated, as one would expect if the method is working. And then the big plot on the right is the Miranda Eve data versus Peter Cook. And here we expect a great aunt, great nephew level relatedness to be 12.5% identical by descent. And that's almost dead on exactly what we see genome wide here. So that answered the question who this girl was. She was Edith Cook. She was reburied with her family with a tombstone that has the right name on it. And I had the weird pleasure of going to a very strange funeral for a girl who no one knew and had known for a century. But it was interesting. It was almost, you know, uplifting in a way. The Knights of Columbus volunteered to help make this happen. The girl was found with a cross, a flower cross on her, and the Knights of Columbus said, we recognize her as Christian, and we will take care of the burial. And very, very nice thing that was done there. And I thought at that point then, this was, you know, an awesome thing. It may be the last time ever that my work is literally, the results are etched in stone. And that was a fun project and a fun thing to work on. Then I got a call from this lady, Barbara Ray Venter. You may know her as the genealogist who solved the Golden State Killer case. She called and said, I saw that on the news. She happens to live down in Monterey. She said in typical Barbara Ray Venter fashion, that was cute. Do you want to do real work now? We can apply this in forensics to catch killers if you're interested. And I, you know, said, yeah, that sounds, you know, awesome. Of course, let's do that. I think this is this point where I can probably stop and do some Q&A if you guys want to do that. This is the end of chapter 1 of the talk.
Yeah, thank you, Ed. Let's see. Let me check the questions. Let's see. First question. How can the girl look so close to life after more than a century?
Yeah, that's a very good question. The casket is the real key there. It was evidently fashionable, especially amongst families of means at that time, to bury folks in very fancy caskets that were airtight. They had, you know, rubber gaskets on them that would seal. And I think that's probably what's behind it. The girl was 150 years old when she was disinterred. It was pretty amazing. And obviously I wasn't present for any of that, but I was told that when they first opened the casket was open for about a day when the sampling was done, and then it was reshut. But during that day, the mold that you see on her face, that's when that mold grew, that wasn't even there when they first opened the casket.
Right. So how many years back is considered ancient DNA? It's, you know, it's not what one thinks of as ancient, right? You think of thousands, tens of thousands of years, hundreds of millions.
Right. There's a fine line between old and ancient. And as a 48-year-old, I think the line is somewhere north of 48. I'm happy to be old, but not ancient. Kidding aside, the ancient DNA, the field of ancient DNA is really describing any sample that is old enough to be chemically damaged. And in terms of years, that process can happen very quickly, or it can play out over long times. In our lab, my partner, Beth Shapiro, they, I think, currently hold the record for the oldest DNA ever extracted. It's close to 1 million years old, but that is from a very special sample that of an animal that died in the Arctic, died in a frozen environment, in a place that has been frozen since the animal died. So was fortunate enough to more or less die in a freezer and never be unthawed. Nevertheless, it was very difficult sample to work with, highly fragmented, and you know, a challenge to retrieve any DNA from that sample. So the back end of ancient goes out as long as DNA can preserve, when in very special circumstances, up to about 1 million years. The narrow end of it, when does old turn into ancient? Well, in the field of DNA, it's really a description of the DNA, not the description of its age.
Well, related to that, you of course, use the hair as the source of the DNA. Why not the bones or the teeth, which seem to be things that, you know, when we think about skeletons, they seem to last?
Yeah, that's a good question. In this case, the motivation was we didn't want to do anything disrespectful to the body at all. It was a difficult thing to imagine what is morally correct in this instance, and what is, you know, what is the right thing to do. And it was pretty clear that doing anything other than taking a lock of hair was just not acceptable by the group. So we didn't have much choice in this instance. As I will get to later here, it turns out that hair is a fantastic vesicle for DNA preservation. And in fact, if I had to choose now between getting DNA out of the hair of a Neanderthal or bone of a Neanderthal, hands down, hair is the way to go. The reason that that's not more common in the field of ancient DNA is hair just doesn't last. It gets blown away. It's hard to identify. You might see a small fragment of hair and wonder whether there's no, you know, morphological trait of a hair that would identify it as an interesting archaic species. Whereas a bone, it to experts, they announce what they are. They announce more or less, you know, whether they're interesting or not. And the shape of the bone tells you this is something that you might be interested in the DNA of. A hair doesn't necessarily do that, and they're hard to find, and they're, you know, they just don't fossilize. They're not something that would be recovered in a typical archaeological examination.
How much of a sample do you actually need? So you talked about not wanting to disturb the corpse, but do you need a significant sample to do that?
Yeah, this is great. It's a great presage to the second part of this. What we had, I showed a picture of it, probably 100 hairs, a few centimeters of 100 hairs, a lock of hair. We used it all, and we got about 13% of the genome. More or less, back then, we sucked. The methods that we were using were incredibly inefficient. And now that we're focused exactly on this problem, we can get about 5-fold coverage, about 500% of the genome from a single hair. And so what we needed back then was more than we took. It would be way more than we would need now would we do this again.
Okay. How do you calculate the expected overlap in genome or related individuals? Or is that something you'll talk about later?
I can talk about that later. I can give I didn't want to get too much into the technical details of that, but the key to this is that if you have we all know we have 2 chromosomes everywhere in our genome. So if they're, one of the pieces of DNA came from mom, one came from dad. And if you're related to someone and you have a segment that's identical by descent, it means that one or the other of these chromosomes is going to be identical, exact same sequence to that person with whom you are related in the segments that are identical by descent. That won't be everywhere, but the challenge is then to find them. What we do is develop a model that tests the likelihood of 3 different things. And these are the 3 different ways you could possibly be related to somebody else. Way 1 is you're not related on either chromosome. Neither one of your chromosomes is related to either one of the other person's chromosomes. That's way number 1. Way number 2 is both of your chromosomes are identical to both of the chromosomes of that other person. That's very rare. That's just kind of full sibs or identical twins, or if we're comparing a sample to, itself, that's a very rare thing. You're not even related that way to your parents, but that does happen sometimes. The third way that you can be related is one chromosome is identical by descent, the other one is not. So those 3 cases cover everything. There are only ways you can be genetically related to somebody else at a particular place in the genome. So what we do is look at the alleles, look at the DNA we have, compare the 2 samples, and ask, as we're going across the genome, are these data matching alleles at a rate that we would expect by the first model, that you're unrelated, or the second model, you share 2 chromosomes, or the third model, you share 1 chromosome. And whenever one of those models jumps out over the other 2, they explain the data better than the other 2. It's easy to kind of, wrap this into a log odds ratio and say, it looks like here you got 1 chromosome identical by descent. And because of the close relationship between Peter Cook and Edith Cook, great aunt, great nephew level, we're talking about tens of millions of base pairs long that are identical by descent. These segments are not like subtle or hard to find. Tens of millions of bases of DNA in a row where one of those chromosomes is exactly the same thing. Actual pretty easy case, as it turns out.
Was it a similar technique that was used to identify Richard III, who was dug up from a parking lot in London a few years ago?
That I don't know. There are other ways of doing identifying things genetically, and we'll get into that when I talk about traditional forensics. What has been done up to now for DNA-based identification. Okay. I suspect that it was an STR analysis, but I don't know for sure.
All right. Does the sex of the victim make it easier or harder to trace ancestry?
Really no difference. Most of the genome is the same, whether one is genetically male or genetically female. We have about 3 billion bases of DNA in the genome, and the X chromosome, I think, is about 150 million base pairs long, and the Y chromosome is tiny. It's about 40 million. So we're just talking about a few % of the genome that would be different in any way if you're male or female, genetically male or female. Most of it is what we call the autosomes, which genetically male and female carry 2 copies of, get 1 from mom, 1 from dad. The genetics of that work the same way.
And let's see, did you go back and ask the question what the cause of death was, and would you be able to do that from this kind of sampling technique?
Yes. One of our team members, Dr. Eerkens from Davis, looked at some rare isotopes and was able somehow, this is outside of my real field of expertise, but somehow to determine that her proximate cause of death was starvation, but it's likely that she suffered from something else that was leading to that starvation. That summer, the summer of 1876 in San Francisco, was a bad summer for malaria and for some other things, and it was a high mortality rate at that time based on the analysis that he has done. We wouldn't be able to see anything directly from the DNA about that, but he did some isotope analysis from the hair and was able to make that conclusion. I'm selling his work short. It's not my field so much, but people were looking into that. What was odd was, when we finally figured out who she was, you can go back and look at the newspaper of the day of her funeral, and there's the announcement, and the family will do a reception. And it's a very odd thing that this little girl who died tragically, but was mourned in her time. It was, everything went as it should, other than a kid dying, which is, terrible and should never happen. But given that happened, there was really nothing remarkable about her other than they missed her grave when they were moving the bodies.
Yeah. And last question for now. And I think this may have been inspired by my question about Richard III, who was identified by by comparison with a living relative. Obviously, Richard III lived a lot longer ago. So as generations go by, how much do the genes drift away from each other? Is there a limit in time where you couldn't tell if people were related?
Yes, we will get into this in some detail if your patience is there, and I can go quickly. Great. The big picture is the signal is lost by half every generation. You share half your DNA 1 generation ago, a quarter 2 generations ago, an eighth 3 generations ago, and it just keeps going on like that, plus or minus some stochastic variation, which is based on the vagaries of recombination and exactly where that happens. So it's easy within a few generations, and then it gets really, really, really hard.
Okay, so let's return to your presentation.
Fantastic. Close that. So this is a slide to just introduce you to the workhorse method in DNA-based forensics for decades. This is the PCR-based method. PCR is a method that just amplifies a particular segment of DNA. And the way that personal identity is established by DNA analysis, a genetic fingerprint or a DNA fingerprint, is by this method. The little blue things up here are meant to symbolize PCR primers. PCR stands for polymerase chain reaction. It's a decades-old technique for just amplifying a little segment of DNA. And the little segments of DNA that are amplified for this STR analysis, for determining, if that was the bad guy, does this DNA match what we found at the crime scene? They're chosen because they have these repeats, like shown here, AATG, AATG, AATG. Turns out those regions of the genome that have these little repeats, they're slippery when the DNA is copied, and they mutate a lot, they change a lot. So there's enormous variation from person to person in these particular segments of the genome. You can amplify up a few of these and get a signal that is specific to one particular person. You can make this data, and it will have come from exactly one person and is very much unlikely to match anybody else. This is the DNA fingerprint that is used by law enforcement, has been used for decades. Arrestees, violent offenders, the rules kind of differ from place to place, but if DNA is collected from someone and then their DNA is put in a database, this is the assay that's done. They don't sequence the whole genome. They do this PCR, look at how long these repeats are, and say, here's what your DNA looks like. And if we ever see DNA from a crime scene and it looks the same way at these markers, then that's you. This is the way DNA-based forensics was done forever. These are the CODIS markers. The data is kept in the CODIS database, which is run by the FBI. They control the access to it. This is DNA-based forensics. The limitation of this is that they require about 10-fold coverage of DNA. Your sample has to have the genome 10 times over to have 95% chance of seeing both alleles, the one you got from mom and dad, at all of the core markers, all of the core 16 markers. And the other bad thing is that you have to, because of this assay, you have to have these PCR primers flank the region that you want to amplify. the DNA itself needs to be big enough that one of the PCR primers can fit on one piece of the DNA at one end and the other can fit here. If the DNA is highly fragmented and you don't have an intact fragment that spans all of this, the method just fails. And as I showed you before, the DNA in hair that you cut off your head right now, this is how long it is. This is recent work that soon to be published showing the fragment lengths of DNA from the nuclear genome and from the mitochondrial genome that you get out of fresh cut hair, either head hair or pubic hair, doesn't matter, looks the same. And the point here is that rarely are these fragments longer than about 100 base pairs long, about 100 ACs, Gs, or Ts. Mostly they're 40 to 80 base pairs long, very short fragments. This is because as the hair is growing out of your hair follicle, the last stage of that development of making the physical substrate of hair is a process called apoptosis, programmed cell death. Endonucleases, little things that chop up DNA, come on, and that's basically the cell turning out the lights. It's going to chop up your DNA, and that cell stops being a cell. By that point, it's filled up with keratin. It's made cross links with the nearby cells, and it's not a cell anymore. It's a physical part of your hair, and it sloughs off. So plenty of DNA in it. All of the DNA that was in those cells, it just gets chopped up into little pieces. This is how long the fragments need to be in order to get a PCR product from each of the CODIS markers. Each marker is shown here, and how long that the piece of DNA needs to be is shown here for the several different kits, different vendors that sell this assay. And as you can see, DNA is typically down here in this length, and it needs to be up here in this length for this assay to work. So this is one of these real facepalm moments in science where for decades, you'll still see it on TV shows, today, that when you try to do a genetic profile from hair, from the DNA you get out of hair, you can't get it. The assay fails. And the conclusion has been that there's no nuclear DNA in hair. The truth is there's plenty of nuclear DNA in hair. It's just too short for the only assay they ever do to work. And it is exactly as kind of obvious and, oh my gosh, as it is stated here. Really, really crazy, terrible. We could have been doing DNA things with hair forever. Just the things we were trying required big pieces of DNA, and they're all short. So a grad student in my group, fantastically talented guy, Josh Kapp, and I invented this method for taking small amounts of fragmented DNA, originally for ancient DNA stuff, but it works great for DNA in hair, and attaching the sequencing adapters, which are necessary to learn the sequence of DNA. I won't get into the details of this, just suffice it to say, it works really, really well. It's designed to be extremely efficient at converting small amounts of fragmented DNA into a form that you can then put on a sequencer and learn the sequence of it. This is the kind of founding technology for the Claret and Astrea Forensics companies, using this for biomedical purposes and for forensic purposes. This slide just shows that it's way more efficient. The Santa Cruz method is more efficient than other methods at converting DNA into a form that you can sequence. And now we've started applying this to casework. What we see is that in real cases, the DNA, or at least the ones we get, the hardest of the hard stuff, they're decades old. The DNA fragments are typically even shorter than you see in fresh hair, kind of like the Miranda Eve. Not quite as bad as Miranda Eve, but DNA from the 70s and 80s is 30 or 50 base pairs long. It continues to fragment inside the hair. So with this method, what can we do with the DNA that comes out of a single hair? Well, we can typically get about 100 picograms of DNA out of single hair. And with this more improved, more efficient method, we can get about 5-fold genome coverage from that. So see every place in the genome on average 5 times. Some places we see less, sometimes more, but on average about 5 times. And so now we can ask, what can you do with that? What can you do with the DNA out of a single hair? The short answer is we can make a DNA file for genetic genealogy, for doing something like relative finding, like is done at 23andMe or Ancestry. The longer answer, I will go into here, trying to be, like I'm down to about 8 minutes here. I'm gonna try to go fast. This is a slide just to introduce relative finding and how this works through genetics. If you imagine that you're a member of the current generation, I used to think of myself as a member of the current generation, less and less as time goes on, but a member of the current generation would have 2 chromosomes, or 2 copies of each chromosome. So this is just some chromosome. One of them, this person got from his dad, and the other one he got from his mom. Simple enough. The reason genetics is so interesting and relative finding is possible is that this chromosome that you get from your dad and this chromosome that you get from your mom, they didn't exist in your dad or your mom. This configuration of parts was in no cell of your dad except for that one single sperm that was to become you. Likewise, for the chromosome you got from your mom, doesn't look like this in any of her cells, just in the egg that was to be fertilized by that one sperm that would become you. And that process of making this chromosome happens by what we call recombination. Your dad took the chromosome he got from his dad and the chromosome he got from his mom, and at some more or less random place stitches his mom's chromosome together to his dad's chromosome at this random place. That will become that sperm, that will become you. For your brother or your sister, this recombination will happen in a different place. It's random. There are a gazillion different places it could happen along these stretches of DNA that are hundreds of millions of base pairs long. And each one of these germ cells, sperm and egg, then is very different. Therefore, what you get, the DNA that you have inherited, you can track that back through generations. If you just ask the question, what specific person did I get this DNA from? You could track a particular place, like this yellow region, for example. That piece of DNA you got from your father. So 1 generation ago, the answer to the question, who gave me this DNA, was this specific person, that one you called dad. If you want to ask 2 generations ago, who gave me that DNA, it's not dad anymore, it's dad's dad. Dad was 1 generation ago, dad's dad is 2 generations ago. What about 3 generations ago? Well, if the female chromosome is on the bottom, as I have it in this diagram, this piece of DNA came from dad's mom. That's who you got it from. So every generation that you go back, every place in your genome came from one and only one person at that particular depth in your genealogy. So if you ask, at this place in my genome, how am I related to my dad's mom? The answer is you're not. You're not related to your dad's mom at all. You got none of that DNA from your dad's mom. You got all of it from your dad's dad. Some other place you might have gotten from your dad's mom, but that place came all from dad's dad. Genetics in this way, it is a winner-take-all proposition. Any particular place in the genome, one person in your genealogy gave you that DNA, everybody else gave you nothing at that place. Some other place, they might be the winner, but there, they wouldn't be the winner. There's only one winner at each place. So anybody that you could then identify as sharing a bit of this DNA, in this case, that segment came from dad's mom's mom, anybody who shares that must also be related to dad's mom's mom, or somebody who's even further back in the genealogy who would have gotten that segment. So this is, in this way, as we go back in time, each segment of our genome could be co-inherited in multiple people. And if we can identify that co-inheritance based on the differences in DNA that are present, then you can identify relatives. You can see from this process that as you go back in time, the segments that will be co-inherited are shorter and shorter and shorter. That and the total amount of DNA shared will be proportional to how closely related you are. Okay, so that's kind of the big picture of how this works. This is just an excuse to show a picture of my adorable son, but in practice, it kind of looks like this. You can label the segments. It came from dad's dad, dad's mom, etc. Go backwards in time, you could label these segments with the actual people who we know existed. That's what the genealogists do. And go back in time and do this. Okay, so what we do now with DNA from a single hair, there are giant public databases that catalog more or less all human genetic variation. This is kind of the most important one, this 1,000 Genomes public project. They sequence the genomes of 2,000 individuals from all over the world, and from that effort learned all human genetic variation down to 1% worldwide frequency. So there's no genetic variation that exists in more than 1% of the people of the world that's not known to this database. It's all there in this publicly available catalog, which is extremely useful for us, having this catalog. I'm going to skip that. The data that we ultimately want to converge on are genotype files that can be compared to genotype files of other individuals. So the genotype sites that we want to learn the DNA of, they need to match what's in other databases that are going to be searched. So there's the 23andMe, Ancestry database, Family Tree DNA. We don't search those databases. We search databases of customers of these databases who have voluntarily extracted their data and put them in an available database. 23andMe and Ancestry are very proud of the fact that they do not allow law enforcement access. And you can go on their terms and conditions website and see them bragging about the number of times the police have tried to get in and the number of times they have gotten in via legal methods, via subpoena, and the number of times they have gotten in, which is 0. They're very, very proud of that fact. It's a threat to their business model. They don't want law enforcement in their databases, but there's a method. Individuals who are customers, they can download their data. It's their data. Put it in these open databases and say, yes, if this is useful for people, come and search it. That's where the data comes from that we're going to search. And we want to call these sites that are in all those databases. We've developed some technology that I sadly won't have much time to get into, but it uses this data all across the genome to call exactly the sites that we want to know on these genotype arrays. And we can now, with about 2-fold coverage of the genome, we can call genotypes that are accurate enough for doing this relative finding in these publicly available databases. This is from a test sample where we take data that is generated by Ancestry using that technology, just a customer of Ancestry DNA, and search this database. A, B, C, D, and E are the 5 closest relatives of this test person in that database. And the x-axis, the centimorgan match, is how closely they're related, how much of their genome they share as identical by descent. That's in blue. What's in yellow is what we get from a single hair. If we make a genotype file, search the same database, same top 5 relatives, almost exactly the same degree of relatedness in terms of how much of the genome that are shared. So what this allows us to do is say, if we can get a single hair, we can get enough DNA out efficiently enough that we can make this genotype file. I can't tell you who it is, but I can give you this genotype file and you might be able to find some of their relatives if that's useful in a law enforcement setting. So overview, we take hair, bone, whatever, and wind up with this GEDmatch or family tree DNA compatible file. It's a very simple thing in practice. It's just a text file that has all of the positions of these variable sites, what chromosome they're on, what position in that chromosome, and then the 2 alleles that we observe. This is kind of where we stop. This is where our technology ends. We make this genotype file and pass this off to our law enforcement partners, who then do what we consider to be the police work part of it: search the databases, identify relatives for investigative leads, and use that in investigations. Just reiterate, you can go on and see the 23andMe scoreboard and the Ancestry DNA scoreboard. They do not search these databases. They cannot. They've tried by subpoena to get access, and 23andMe and Ancestry say, no way, no how, ain't gonna happen. We've got billion-dollar businesses here that would evaporate overnight if we ever let you in. We're gonna spend any amount of money on lawyers to keep you out, and they're undefeated in that effort. What the police can do, though, is that customers of 23andMe or Ancestry can take their own data, which belongs to them, it's their DNA data, download it, then submit it to GEDmatch or Family Tree DNA, these two open databases where people can voluntarily put their genotype information and then opt in for law enforcement if they choose to do that. That's what that website looks like. I'll finish up just by blowing through, some cases that we've solved. This was a terrible guy. This was on 2020. We did some filming at Santa Cruz. That was a lot of fun. We identified the victims in this case. These were 4 women who were found in barrels who were killed by this guy. And he was a real piece of work, a drifter, and had a bunch of different assumed identities, and killed a lot of people. He was a serial killer, but these women, they knew they were victims of him, but they didn't know who they were. We identified them, which was very useful for their families. This guy was a serial rapist, murderer, kind of similar MO to Golden State Killer, all over kind of the Mountain West. Turns out he was a long haul trucker, and they never really could put the pieces together, but with some really old samples back from the 90s, we're able to figure out who this guy was, and he's been arrested and since confessed, and is doing his time now. Another Jane Doe case that we did that we're very proud of is from hair from an exhumed body. A girl was killed long time ago, back in the 70s, and before the whole forensic genetic genealogy Golden State Killer craze happened, they were trying it with this Jane Doe case, and nobody could ever get anything to work with it. We got the sample. Barbara Ray Venter said, you guys try this, and we're able to identify her as Elizabeth Ann Roberts. She was a rape murder victim. It turns out it was so hard because she was adopted. So the whole genealogy thing is a little bit weird. Her birth family died in a car accident shortly after she was murdered. Of course, no one knew that was her birth family. She was a runaway, and her adopted family had filed a missing persons report, but some clerical error kept that from staying in the missing persons database. So she wasn't even known to be missing through the normal channels. But anyway, genealogy figured it out, and we're able to identify her. This work has been in the newspaper if you want to read that. And it's actually a very good article by Heather Murphy describing this. And I'll finish there just saying that this has been an incredibly fun thing to work on. Santa Cruz is a great place to do this kind of really blue sky application of technology for good. And it's been fun to watch this field grow and change, and it's very much still in flux now. The DOJ has weighed in on some guidelines about when this should be done. And of course, the wisdom of Yogi Berra. It's hard to know here what the future holds. And I will finish up with that. Hopefully I didn't go over too long.
Thank you so much, Ed. That was really, really great. Very, very interesting. You did pick up the pace there, but there was actually not much need to do that. We have some good amount of time for questions. But
okay, good.
But that's great because we have a lot of questions we can ask. You kind of ended there with what is touching upon some issues of ethics around this, and there are many different sides to the ethics question. You mentioned one earlier with respect to disturbing a corpse. There's ethics questions around people entering their DNA into databases. There's ethics around police access to that information. So can you spend a little bit more time sort of walking us through some of these ethical issues and how maybe you personally feel about about them?
Sure, happy to do that. And it's a very deep waters, and we'll just start by saying that it's an area where reasonable people can disagree, and I don't claim to have any superior ability to navigate this space compared to anybody else, but I'm happy to say what my thoughts are, but not in an attempt to make people think like me at all. Would be welcome to have the discussion about this, especially with people that disagree. What makes it difficult, I think, is that on the one hand, you imagine that the ownership of one's DNA or the information in one's genome, there's nothing more personal than your own DNA. And if one is to own that and to control what happens to it, try to imagine who does own that and who should make that control decision. Of course, it has to be the person whose DNA we're talking about. That seems like rock solid proposition to start everything else around. Nothing should violate that. No government or company or agency should ever be able to tell me what I can and can't do with my DNA. What makes this so difficult is that my DNA that I consider to be so personal to me is actually shared with lots of other people, and is in fact, in a lot of ways, not at all personal to me. One could imagine that my DNA is up in the GEDmatch database. I don't personally have a qualm with that, but I recognize that in me making that decision, I've also made a decision for my brother and my sister and my parents and my cousins and lots of people who are related to me. And I didn't ask them. I didn't, all of the permissions that one might want to get before making a decision that might impact somebody's life. I didn't do that. And I'm happy to talk about, why that is. Mainly it's a logistic issue. We'd be talking about thousands of people, but that's what makes this hard. That inviolate premise that my DNA is mine and I get to decide is actually a double-edged sword. And if someone else has that exact same attitude, then they can easily make a decision for you and cut you out of the loop entirely. I think that's the kind of quandary behind all of this. The other interesting ethical aspects to this, the DOJ has finally weighed in on this. Forever with the CODIS database, the access to that database and what samples are in it were 100% under the control of the FBI. And that is, you hear about, security breaches and data breaches all the time, and it seems like everybody has been hacked. That database has never been hacked. The FBI has that on absolute lockdown and controls that. Knock on wood, it has never happened. And they come up with rules of access because they are the government, and the rules are all with kind of an eye toward what should the government be able to do with this DNA information? Who should have access? Under what conditions should they have access? What's been interesting to see with this new forensic genetic genealogy is that it's completely outside the government. There's not a government database. There is a spontaneous user-driven database of people who say, I want to put my data in this database. The history of that database is actually kind of interesting. GEDmatch was a user community long before DNA of people who are interested in genealogy. They wanted to research their family history and figure out, who their cousins are so they can do their family trees. And it was only later when DNA, came along as a tool that could assist this, that they bolted on a little tool to let you download your DNA from 23andMe or Ancestry or whatever. They were frustrated because if they want to find their cousins and you're a customer 23andMe and your cousin was a customer of Ancestry, there's no way to find that relationship. So they built this little database so you could put your data in there and find the cousins. So it's genealogists, hobbyists. Later, visionaries like Steve Kramer at the FBI and Barbara Ray Venter said, we could use this to figure out who the Golden State Killer is, or try to do that. We can get plenty of DNA, left plenty of DNA at the crime scenes. We can figure out who his relatives are. That might actually help. It turns out it helped like crazy. It was great. But that database, never did they ask the FBI or ask the government, can I have access to the database? It wasn't owned by the government. So all of the normal things that you might say, look, government, you got to lock this down. Look, the cat's out of the bag. This is done. It's people who are doing this of their own volition with their own DNA. So that's not really an ethical response to it, but it's definitely an important practical consideration that it's hard to imagine how to undo this now if that were deemed the right thing to do.
Yeah, well, it seems to be certainly the case that states are moving towards, I believe it's states are moving towards the policy of whenever there is any kind of arrest, whether you're found guilty or not, your DNA is taken, a sample is taken, and it's placed in a database. That's of course got interesting implications for really who owns your DNA. The government is deciding that it will take it upon an arrest, not even a conviction. And that you could also ask the question, well, who is it that gets arrested? And how is that biasing the database information that we have?
Yes, there is a well-known bias in the CODIS database, for various reasons, and it brings up issues of justice that are deep and hard and important, and we're thinking about for sure. The forensic genetic genealogy databases, they have a very, very different demographic skew. It's folks who, for whatever reason, were interested in searching historical records and other things to research their family tree. And the facts of the demographics of the folks who have done that make this, it kind of inverts it a lot, which is kind of an interesting thing to contemplate. But in any case, the point you make about folks who were arrested but not convicted, having their DNA then in any kind of database, I think a reasonable person could say that's not right. It shouldn't be that way, and it's a little far afield from what we're doing. We don't ever search the government databases or interact with them in any way. But you're right. That is, I'm against that. What can I say?
Yeah, thank you. There's also, I suppose, other occasions in which the DNA samples are taken and used, but this sort of line between where the government makes the decision, a company makes a decision, the individual makes a decision is really one that I think as a society we're actually going to have to explore. But it kind of begs the question, given all the science, you're talking about you're very complicated, very cutting edge science, and we have police investigators, we have judges, we have juries that somehow have to walk through this and to some extent understand that. How are we going to prepare our society to be able to answer questions or to render judgments based on this kind of scientific evidence? I mean, are they prepared? Is our society prepared? Is the general public prepared to be a member of a jury, or even a judge?
Step by step, to answer the question, if it required really having understanding of this deep enough to be critical of it and discriminating, I'm not sure. Probably no. The DOJ guidelines, as they're written now, say that forensic genetic genealogy, this technology, can only be used to generate investigative leads. It cannot be used in any prosecution. By DOJ guidelines right now, none of this could ever be used to convict anybody. It's a really strange thing, and we've seen this play out in case after case where the police, law enforcement come to us and want to know who did it, and then they'll get other evidence to prosecute that individual, but they don't know who did it. And for the kind of typical cases like Golden State Killer, where they've got a CODIS profile from lots of samples. That's how they knew it was a single individual, this single genetic profile over and over and over again, same MO, same everything. They just don't know who that individual is. If you could tell them who that individual is, they could then go compel a sample, take a discarded sample, do whatever, get other data that says now, yeah, you match this Golden State Killer CODIS profile. And the only thing that was missing is who the bad guy was. And that's what it can be used for now. That's how it is used in practice. But the evidence itself is never presented to a jury, and no jury's ever demanded that they understand it and put somebody in jail or not based on their understanding of this. That doesn't happen. The day is likely coming when that will happen, and we can do our parts. We can have Kraw lectures and talk about this, and do as much outreach as possible. I've been on TV many times now trying to discuss this. It's hilarious what the producers will choose to include and not include. Nobody wants to watch a TV show about the boring science part. They want to hear about the crime and how the crime was solved. Understandable, that's more interesting somehow. But keep trying, keep trying to explain as best one can. Not only what is possible, but what's not possible, and what are the downsides? How does this go sideways? And I'm more hopeful maybe than the average person. I think in general we've got responsible, smart people in lots of places who can make decisions, and as long as we don't try to get ahead of ourselves, then this can be done in a way that is more good than not.
We have a question. Is obtaining DNA sample different than fingerprint samples? So fingerprint samples is a technique that's been utilized for over 100 years. We kind of, there's kind of a standard practice around it. Is it complicated? Is it different? Is it easy?
I don't know so much about collecting fingerprints, honestly. I know that is, a commonly used forensic technique, but it's not something that we've ever been asked to do, nor do I have any knowledge of it. There's a field of forensic DNA called touch DNA, trying to get DNA off surfaces that have been touched. Kind of the next holy grail of DNA-based forensics, and it's something that we are working on developing technology for. But the actual lines of the uniqueness of a fingerprint in the traditional sense, that's not something I really know anything about.
Well, it's related to another question, which is are there ways of masking the DNA, in the sense that you hear about criminals who use acid to remove their fingerprint, or you wear gloves when you commit the crime, but is there a corresponding concept in the DNA world?
Yeah, you know,
Even chemically, I mean, you know, to be more
Sure,
sophisticated than
I've-
wearing a hat.
you know, been involved in some, you know, red hat exercises and like there, of course, yeah. You know, what is difficult for us is small amounts of DNA. We're, you know, way better than we used to be, but it's still hard. Touch DNA, other things where there's tiny amounts of DNA. Mixtures are hard. If you've got DNA from multiple sources, it's a very difficult thing to then figure out who either one of those contributors were. You know, given all of that, I think the smart criminal of tomorrow could come up with some ways to make life very hard for the DNA-based forensic scientists. For example, if you had a spray bottle that had DNA extract from 100 different people and you just sprayed that before you left the crime scene, I'm not sure that would be such a giant mess. Probably would never be able to, but that's why we love hair so much. Hair is an intact, desiccated biological unit that always derives from a single individual. You don't ever get a hair that came from two people. And it, because it is impermeable to most aqueous solution, you can wash the crap out of the outside. Our standard procedure is to do a mild wash in a dilute bleach solution. So really get everything off the exterior, then enzymatically dissolve the hair and release the DNA. It's a perfect little vesicle for enough DNA from exactly one person. But other kinds of forensic samples, blood and semen and fluid, any kind of thing like that, really forward-thinking bad guy could pollute those in a way that would be hard to unpollute.
Yeah. Well, again, related, if you take two samples from the same person, say two pieces of hair, run the analysis twice using those two different samples, would the results from the analyses be perfectly similar, or is there noise in the analysis?
Yeah, there always is. It depends on what one means by the analysis. There are data that are collected and then interpreted all the way from the sample to the genotype file at the end to the relatives who are found. This is something that we're working on campus right now, stress testing this. The answer with the DNA, the raw data that's collected, that would be it would have lots of differences. The little fragments that are present and collected and converted, they're just a random sampling from the genome, and you would be sampling different things. It would be like saying I take two die, six-sided die, the same six things, and I roll them both 100 times. Do these, and then ask do these die have the same characteristics? Do they have the same probability of each one of the numbers in there? If you do this enough time, you can make the statistical argument that yes, they do, but you're not going to get the same sequence of numbers. It becomes a probability thing. So what we are trying to do is infer the genotype from this random sequence data using methods that I didn't really get a chance to explore. That part does have some stochasticity. It does, there are p-values associated with each one of these calls. What seems to be the case though in practice is that some small amount of stochastic error in that does not impact the ability to find relatives. The identity by descent segments that we're finding, they span thousands and thousands of these variable sites. And it becomes very easy to say either you share a chromosome with this person or you don't, if you're looking for these really long stretches. And of course, we're only ever looking for the really long stretches, the second or third cousin level relative. Trying to find someone's 10th cousin who's related by a common ancestor 500 years ago is never going to be a useful investigative leads. It's only the closer relatives that are a priori easier to find and less prone to the statistical fluctuation and the genotype calling that law enforcement's interested in anyway.
Fascinating. Well, Ed, we're about out of time, and I want to thank you on behalf of the audience. We didn't get to all the questions that came in, and comments too. I think people are very interested and engaged with this topic. I noticed a number of people who would like to contribute to a conversation about this, and I wonder if there's a forum which we could put together in which the community can come and discuss with you and other experts in this area to talk about the science of it, but also the ethics and the practicality. And I hope we can do that, and I hope you'd be interested in doing that.
Of course.
Great. All right. Thank you very much, Ed. And I'll turn it back to George Kraw right now.
I'm having a little problem here with my video, but I want to thank you, Alex and Ed, for that fascinating presentation. Please join us for our next Kraw Lecture, May 19th, Precision Genomics, bringing research into the clinic for children and cancer. Good night, everybody.